Hey everyone, Dr. Jack Gordy here, and today I'm going to be taking you through a paper, and this paper covers the AstraZeneca vaccine in a clinical trial. And it's a very interesting paper, so let's strap in. So, first of all, the paper was published in Nature Medicine, which is a fantastic journal, and the paper covers T-cell and antibody responses induced by a single dose of the AstraZeneca vaccine in a phase 1, phase 2 clinical trial. The AstraZeneca vaccine is a Chimpanzee adenovirus vaccine. So that's what the CHAD stands for. And adenovirus is a different kind of virus. Remember, it's not a coronavirus. And it's been genetically modified to do two things. One, it's been genetically modified so it cannot replicate. So now it is a live virus, but it can't replicate in your body. And two, it contains a gene for the SARS-CoV-2 spike protein, right? So how does this work, right? Well, we inject it into our body. It can't replicate, but it can do what viruses do, which is insert its genetic material into the host cell. And so these adenoviruses, these chimpanzee adenoviruses, will insert their genetic material into our host cells, which is us, and our cells will now start from that RNA, producing spike protein. So now the cells that have been infected by the adenovirus, instead of creating more adenoviruses, they will just create spike proteins. Those spike proteins will get expressed and displayed on the surface of antigen-presenting cells, and then those antigen-presenting cells will display it to T cells, and the T cells will activate the B cells, and then we will end up with a great adaptive immune response, and we'll end up with memory B cells and memory T cells that will remember SARS-CoV-2 and the spike protein. Fantastic. Why did they choose a chimpanzee virus? Well, they chose the chimpanzee virus because humans are exposed to adenoviruses all the time. And they cause a certain percentage of the common cold, and they also cause gut problems, for example. So in our blood, patients may have antibodies for human adenoviruses. And it's those antibodies may alter the way we respond to the virus. So rather than the virus getting into cells, for example, they might get coated in the antibodies for adenoviruses that are already in our blood, and that will prevent the adenovirus from entering the host cell to deliver its genetic material. So we don't want to be injecting a viral vector for SARS-CoV-2 that we might have already seen, because it won't do its job as well, because we might already have antibodies for it. Because humans have not seen this chimpanzee adenovirus, we won't have antibodies floating around for the adenovirus in our blood. So therefore, it can do what adenoviruses do, insert its genome into our host cells, so we start expressing spike proteins. So that's the deal with this vaccine. Let's jump into it just quickly. Clinical trials are loosely broken up into 3 phases. There is actually a 4th phase, which has been very important for the AstraZeneca vaccine. So phase 1 is safety. We take a small number of not sick patients and we give them the drug and make sure that it doesn't induce horrible side effects. Phase 2, we look at a proxy measure of efficacy. So we don't have enough in our study to see if our vaccines or our drugs actually work. But we do have enough in our study to see if the supposed mechanism by which they would work is occurring, right? So an example might be, say we want to know if this drug is therapeutic in stroke, and the drug is supposed to be an anti-inflammatory. The first thing that we might do is just test in a small bunch of stroke patients to see if we've reduced the inflammation. The next step we would do is, did we improve outcomes? Did we improve mobility, cognitive outcomes? Did we reduce the amount of brain tissue that died? And because that's so hard, you need so many more patients. So you want to make sure that the drug is actually doing what's on the label first, and then you want to do a big trial which involves lots of people. Now, the phase 3 is a clinically relevant efficacy measure. Does the drug work? Does the vaccine work? Does what the intervention actually work? Now, I mentioned phase 4. Phase 3, you get approval and the drug gets rolled out, or the vaccine gets rolled out. Phase 4 is we watch really closely. And that's because sometimes there are side effects that might be 1 in a million. And this is actually what's happened with the AstraZeneca vaccine. We've found that it can cause clots, and those clots can cause death. And that might occur at about a rate of 1 in a million. Now, because To detect that, you would need to run a phase 3 clinical trial of tens of millions of people, which would literally cost trillions of dollars, probably, given that it costs around about $2 billion to run a phase 3 clinical trial on 40,000 people. Imagine what it's going to cost on tens of millions of people. It's going to cost a heck ton of money and take a prohibitive amount of time. We would never get any drugs approved, basically. Because of that, we just roll the drug out and we say, obviously, we didn't see anything bad in 40,000 patients, so we're pretty darn sure this is safe. But we cannot detect something that's 1 in a million, right? We need tens of millions to detect something that's 1 in a million. And so now that we've rolled the vaccine out to tens of millions of people, we watch really carefully. And what we found was there was this tiny, tiny, tiny fraction of people that had this very negative side effect. So we've paused it, or we've avoided those fraction of people, because it seems to be happening mostly in women under the age of 50, these blood clots. So if we don't give it to women under the age of 50, we probably aren't going to get that side effect. And so, and that's completely unexpected, right? But what it shows is that the system is working. We are doing that important phase 4 trial, and we are watching incredibly closely to make sure we're not making any mistakes, which is fantastic. Anyway, so this is a phase 1, phase 2. Now, this is because we were in the middle of a pandemic. We decided to merge those phases. We know from previous research that adenovirus vaccines are safe, right? So we thought, let's just combine this phase 1, phase 2 clinical trial, because we don't have 6 months to run the phase trial, 6 months to raise the phase 1 trial, 6 months to do the phase 2 trial. Meanwhile, tens of millions of people, no, millions of people are dying from the pandemic, right? So they combined them into a phase 1, phase 2 trial. Was there any negative side effects? And did we get a proxy measure of efficacy? Now, what do we mean by that? We mean something that approximates efficacy but isn't efficacy. And this is where this part comes into it, T-cell and antibody responses. If the patient is producing memory T-cells and antibodies for SARS-CoV-2, that is a proxy measure that the vaccine is working. We don't know that those antibodies or T-cells will stop infections or reduce symptoms, so we don't know whether the vaccine is actually working, but we know that the point of vaccines is to create memory T-cells, memory B-cells, and circulating antibodies. That is the point of vaccines. So if the vaccine is doing that, that's a really good sign that the proxy measure of efficacy is happening, that these will be efficacious, and so that's a really good sign that we should jump in and do the phase 3 clinical trial. If you gave the vaccine to a small number of people and you didn't get any antibodies and you didn't get any memory T cells, you wouldn't go on to a phase 3 clinical trial. There's no point. It's not doing what's on the label, so we don't need to continue on. So that's the point of this paper. It is the phase 1, phase 2 clinical trial of the AstraZeneca Oxford vaccine. And what we're looking for here is we're trying to get this response here. We don't want a pure Th1 response, because as you saw in my previous video, a pure Th1 response, you won't get the best sort of antibody response that you can get. We don't want a pure Th2 response, because then you're not going to get the cytotoxic T cell memory response going on here. So we want a mixture, probably a little bit leaning to Th1 is what we're looking for. So what did they do in the study? They took healthy adults aged 18 to 55 and they took 88, right? Now, 88 is a heck ton less than 43,000, which is what ended up in the phase 3 clinical trial for this. So you can see that it's worth doing this phase 1, phase 2 clinical trial, because you don't want to spend hundreds of millions or billions of dollars doing a massive phase 3 clinical trial when we can't even get those antibody responses. So we've got 88 individuals here, and they were randomized to receive one of 2 vaccines, right? So they're either going to receive the AstraZeneca vaccine, or they were going to receive meningococcal disease vaccine for a gram-negative bacteria. And this is because you want something to compare against. I actually think this is kind of a random thing to compare against, because it's an antibacterial vaccine response. It's a vaccine for a bacteria, which I think is kind of random. But any vaccine, because of the adjuvants in it, because there's pathogen-associated molecular patterns, are going to cause some response, right? So you don't want to compare a vaccine for SARS-CoV-2 against nothing, because there will be all these sorts of responses going on, inflammatory responses, PAMPs going on, and even if the vaccine doesn't work because of the adjuvants, because of the adenovirus, capsid proteins, and so on and so forth. So we want something relevant that will also cause inflammation and cause an adaptive immune response to compare against. So they chose this vaccine, which is an incredibly safe vaccine. It's 80 to 90% effective. So it's comparing sort of a good vaccine against an unknown vaccine. Let's see how they stack up. Our blood samples were taken prior to the vaccination on the day of vaccination. So day 0. And day 7, day 14, day 28, and day 56 after vaccination. So these patients got poked a lot. They got sampled a lot. And these patients deserve, these participants deserve a badge of honor. You know, they helped in the development of a vaccine that's likely going to be very important in the profile of things. Perhaps not as important as we thought, but still very important, particularly because these vaccines are much cheaper and easier to produce than the Pfizer and Moderna vaccine. Then they do things like a cytokine bead-like assay. It's not quite, it's called a mesoscale assay, but it's very similar to the cytokine bead assay, which I've covered previously, to look at the cytokine profile changes that go on. They also did flow cytometry, looking for B cells, T cell subtypes, natural killer cells, and cell proliferation. They use this Ki-67, which is a label for cell proliferation. And they also did various different kinds of ELISAs, which I've covered in my previous videos, to look for antibodies, the quality of those antibodies, the types of those antibodies, and also the T cell responses going on. Right, whoa, so this is figure 1. Right off the bat, we've got a pretty intense figure here. A lot of different colored splots on axes, which, you know, it's not very obvious. I don't think the average person on the street would have any idea what this is. It looks like a bunch of colored splodges on a page. So let me just quickly do a refresher on what kind of graph this is, and then we can jump into the details on what they found. So this is the results of flow cytometry, right? So flow cytometry, you label cells, run them through the machine, measure the fluorescent levels, and you record those fluorescent levels to look for various markers, right? So here is an example, a very simple example of a flow cytometry plot. On the y-axis, we've got a forward scatter, which is an approximate for size. It's essentially kind of like looking at the shadow of the cell, how much light gets scattered away when you shine a light on the cell, right? So this is an approximate for cell size. And here we've got an antibody with a fluorescent label on it, and this is for CD3. Now, CD3 is part of the T cell receptor response, so it's present on all T cells. And cell size up here, these guys are much bigger than these guys, so these guys might be macrophages, right? They might be a big cell, and these are small cells that are positive for CD3, so these are T cells. So we've got macrophages and T cells. But you notice here that the T cells have different colors, right? That's because we know that there's lots of different kinds of T cells, right? So in this two-dimensional graph, all those T cells are stacked on top of each other. So we can't tell the difference. Which one is which? What kind of T cells are in there? So what we could do is, instead of a 2D graph, we could generate a 3D graph with a Z-axis. And that would look like this. So now we've got Y-axis, X-axis, and a Z-axis going deep into the page here. So we've got a 3D graph. And on this one, we could have fluorescence for using a different fluorescent probe. So this one might be red, this one might be green. And this is for anti-CD8. Now we know CD8 is only present on cytotoxic T cells because CD8 binds to MHC1. And MHC1 is expressed on all cells. So that's where the cytotoxic T cells are. And so we can see that if we go up the page here, these yellow dots are now really high fluorescence on the CD8 label, and they're also high on the CD3 label. These orange dots are very low on the CD8 label and very high on the CD3 label, and these guys have neither CD8 or CD3, right? So now we can see these are probably our cytotoxic T cells. These are our T helper cells, our CD4 positive probably. And these are our macrophages. They're not expressing either CD8 or a T cell receptor. So these are probably, and they're very big. So these are our macrophages, right? Now what we can do is we've got a three-dimensional graph. We can now reduce the dimensionality of it, which is essentially taking the cube and squishing it into a two-dimensional shape. And we do that just by, let's redraw the axis, right? This is a very simplified description of what we're doing when it comes to one of these plots that are called a t-SNE plot. So it's a way of, now we can see on a two-dimensional graph our three cell population. Now these units here, TC1 and TC2, now don't actually mean anything specific, right? They basically mean how distant are these cells from each other, right? They don't mean anything specific like CD8 or MAC or CD3 or cell size. They don't mean anything like that. All they kind of mean is the relative relationship between these cells here. And so now we can see that they're clustering separately. And you can kind of see it's a great way to reduce dimensionality. Now let's imagine we then have another probe, but this is for a different receptor, maybe a CD4. Then we have another probe, maybe it's MHC2. Then we have another probe, maybe it's IL-5 receptor, and so on and so forth. And every time we make it a three-dimensional graph, but then we squish the dimensionality down and we reduce it down to 2D. Now we could end up looking at all our cell populations on one graph. And remember, the units don't mean anything, but just roughly the distance. How different are these cells from each other? So now when we look back at here, we can see what they've done is they've taken all 44 patients and they've taken the flow cytometry results from day 0 and combined it with the flow cytometry results after vaccination, day 7, 14, 56. And they've clustered the cells, and they've done flow cytometry for loads of labels. Labels for natural killer cells, labels for B cells, label for cytotoxic T cells, T helper cells, and so on and so forth. And what we can do is now look at those populations and see what other labels they've got to see what's going on. So now as we go through it, here we can see our B cells here, and this is our post-vaccination B cells in the light green there, kind of the teal, I'd say. And we can see our pre-vaccination guys are on a slightly different location than the B cells post-vaccination. Now remember, location doesn't mean anything specific, but it means that on some of the things that were measured, now lots of things were measured, but on some of the things that were measured, they are now different after post-vaccination compared to pre, because those populations don't completely overlap. They're just different. That's all you know. They're a little bit different. And so here's our CD8 cells. This is post-vaccination in dark blue. And here's our pink CD8 cells prior to vaccination. So you can actually see a lot has changed perhaps on these guys. These guys are perhaps very different over here. These guys are somewhat different on some of the things that were measured during flow cytometry. So what was measured? What could these differences possibly be that caused these shifts in locations of the population? Well, one thing that I measured was Ki-67. Now, Ki-67 measures cell proliferation. Cell proliferation will come, those cells will come up positive for Ki-67. And they also measured IgG. So you can imagine there's not a lot of expression of IgG pre-vaccination, but you're hoping that there'll be a lot of expression of IgG post-vaccination. So let's have a look. Here's our B cell population in the exact same location on the graph there in the blue box there. And we can see now we've got a bit of yellow and we've got a bit of orange. So that's probably what's caused that subtle shift in the B cell population from day 0 to post-vaccination in the location on this graph, is that they're now dividing because some of them are positive in Ki-67. So we're getting proliferation of our B cells, memory cells, and effector cells probably. And we're getting the production of IgG. So that would be effector cells. So that's a good sign that our vaccine is slightly working. I've got to say it's not a lot. This, if we go back to, that's our whole B cell population. Just got a smattering of IgG expression and cell division. Our natural killer cells, though, so I didn't really cover this, but natural killer cells, when stimulated with various cytokines, undergo massive proliferation. Our natural killer cells are massively proliferating there, which is, good, because natural killer cells are good at killing virally infected cells. So that's a good response there. But natural killer cells don't form memory natural killer cells, right? They're an innate immune cell. So it's a little bit of a problem because it's not really what we want the vaccine to be doing. We want the vaccine to be stimulating our adaptive immune system, not our innate immune system. Because our innate immune system, too much, we need a little bit, but we don't want too much, because they don't produce memory cells. Here we go. So that was day 7. Here we have day 14. We've got a bit more proliferation in our B cells. Our natural killer cells are still proliferating because they're still positive for this Ki67. Our B cells here are producing IgG a little bit and a little bit of Ki67. We've got our CD8 T cells. There's a little bit of yellow in there, so they are proliferating. The purple is a different marker for a different thing. It's about whether or not where they came from. But yeah, so we've got a little bit of yellow in there, so we've got a little bit of proliferation of our cytotoxic T cells, and we've got quite a lot of yellow in here, which are our CD4 positive T helper cells. So day 14, we've got a lot of CD4 helper cells. T helper cells proliferating, natural killer cells are still proliferating as well. So in conclusion, we've got some B cell proliferation and IgG expression, we've got lots of natural killer cell proliferation, and we've got lots of T helper cell and some cytotoxic T cell proliferation there, right? So that immediately looks quite good, right? It looks like the vaccines are working. We're getting the kinds of responses we want there. Perhaps we'd want more B cell proliferation, but we're going to jump into that a little bit later. Okay, so up next in the next video, sorry, this was a bit of a long one. We're going to be talking about how do we measure cell proliferation a bit better and the cytokine responses going on. What's causing these changes in cell response?